Hi, this video describes a GWAS, which stands for Genome-wide Association Studies. These fall under the broad category of agnostic studies, meaning studies that test many hypotheses. The main aim here is to see if a certain genetic trait is responsible for a particular disease. Here is an image of the human genome. We know that we have 23 pairs of chromosomes that carry our genetic information and are responsible for various physical traits. Eye color, hair color, height, for example. Zooming in further, on the left side is a single chromosome. The colored bands are called genes. For example, the red-orange pair renders eye color, or the blue pair gives us our height, and so on. Zooming in further to any one of those genes, we should expect to see the base units mentioned on the right at level 0, A, G, C, and T, standing for adenine, guanine, cytosine, and thymine, respectively. You can think of them as alphabets. Together, the way letters make words, the base units make sections of DNA, and those are our words. The structure on level 1 are called base pairs. Those are our words. The DNA is a double helix structure, and hence you can see a railway track-like structure. Usually only one of those two strands is represented while the other is understood because A will always bind with T and C will always bind with G. We must also keep in mind that we inherit one copy of the gene from our mother and one from the father. So let's say the red one came from the father while the orange one came from the mother. To make things simpler, let us just focus on one gene for now. We will denote the red with the sequence GTCCAGCAC and call it capital A. Due to inheritance and mutations over generations, there can be a change. Let's say the third C changed to a G. This being a variant or a different version of the previous one, we will denote it by a small a. These two are said to be alleles of each other. The change that we mentioned is called a SNP, or a single nucleotide polymorphism. Carrying the previous concept to more mutations along the DNA, we now see that not only the C changed to G, but the T changed to G as well. Now that we considered more base pairs, we will change our nomenclature from alleles to haplotypes. If we zoom out from the individual level to a population or a group level data, let's see what happens. Considering only one SNP, there are two possibilities, the capital A and the small a. Some individuals will have inherited both capital A's from mother and father. Some would have inherited both small a versions from mother and father, while some would have got a big A and a little a from mother and father. Let's say we have 5, 7, and 4 such persons respectively, or total 16 people in the group. This is the genotypic frequency, 5, 4, and 7. We can also change the parlance and talk about the allele frequency. Since there are 16 individuals, there will be a pool of 32 genes, since everyone would have got one from the mother and one from the father. And from the pool of 32 genes, there are 17 capital A genes and 15 small a genes, and hence those are the allele frequencies. Now things get a little more interesting. In this group of 16 individuals, we also measure their BMI along with other things like height or weight. And we happen to observe that those 5 people with both capital A have lowest BMI, while people carrying both small a have highest BMI as shown on the right. Now we are developing a data set and a preliminary model. So we are trying to see if a certain gene setting is associated with certain disease or traits, or for something like BMI. We can do regression analyses depending on the outcome. In the previous slide, we saw that BMI, which is a continuous trait, would require something like a linear regression analysis. We will treat genotype, which is the both capital A's, the both small a's, one big A and one small a as a ordinal predictor variable and get an associated slope and its p-value. We could also do a chi-square test, but these will only incorporate two variables at a time. If we wish to adjust for confounders, we can extend the idea of a simple linear regression to a multiple linear regression analysis. Race ethnicity is sometimes considered a confounder in such analysis. An important consideration to note is that we would be doing the regression analysis over and over, probably more than 10,000 times. We would be testing the hypothesis that a SNP is associated with the outcome, for example BMI, multiple times. If we use the same 5% type 1 error rate, then we can easily catch some of the SNPs that are associated, but this would be a false positive, meaning we would detect an association even when none exists. For example, 5% of 10,000 is 500. Are all those 500 SNPs causing the outcome? Probably not, because some of them were caught by error. Hence, we need to do some sort of a correction for multiple testing, for example, a Bonferroni correction. Finally, after all the analysis, most of the research papers will show a plot like the one you're seeing currently. It's called a Manhattan plot. Each dot, although the image is not very clear, is a result from all those simple linear regressions or a multiple linear regression, depicting a p-value compared against the multiple testing adjusted threshold, which usually is accepted as 5 into 10 to the power minus 8. The SNPs that cross the horizontal gray dotted line are the significant regions of DNA responsible for the outcome, which brings us back to the main question that we started as an investigator, is what is the cause of the outcome at the gene level? Here are two variants of such agnostic studies, a GWAS, which is a genome-wide interaction study, or an GWAS, which is epigenome-wide interaction studies, among others. Please let me know in the comments if you liked the video and hit the like button if you enjoyed this.